Okay, thanks a lot. Yes, so the title of the talk was going to be hardware and software for next generation electrophysiology, but Tim did an absolutely fantastic job of presenting the hardware, so one word has gone from the title. What I'd like to talk about is what do we do with the data that it's now possible to record using these technologies. It's a complete revolution in terms of how we're going to do science. So what are we going to do once we've got the data? And specifically, there are two questions. The first one is about pre-processing. So you record this wideband stream of electrical signals from all over the brain. How do you detect the times of the action potentials that generated those potentials? And this is what's called spike sorting. The other question is how to use this data to draw scientific conclusions. We heard a lot about number two yesterday from various people. Today I'd like to instead just focus on number one, the more technical issue of how to detect the times of the action potentials from the very large data streams that are coming in. So this is not a new problem. It's been around for quite a while, and there's a traditional method that has been what people have generally used some variant of for decades when recordings were just made with single wires or with tetrodes, bundles of 4 wires. And it goes through various stages. So first, you would have, in this case, a tetrode, four wires in the brain, and it would record 4 signals. And the point is that any neuron that fires produces an action potential waveform that's detectable on all the channels, but the relative amplitudes on the different channels and the precise waveforms differ between the neurons, which is how you can tell the times of the neurons apart using this. So the traditional analysis pipeline, you'd take your raw data, you'd high-pass filter it to get rid of this slow local field potential signal and keep only the fast action potential signals. You then detect the times of the spikes using something like a threshold crossing. You'd take them all out and align them all together so they sit on top of each other. And here you see, for example, 2 superimposed waveform groups from different cells, which we're going to try and tell apart. The next step is to compress the data, going from these waveforms, which are, say, 32 samples long, to just a few numbers that describe each one. This is normally done with principal component analysis. And it gives you what we call a cluster plot, where you're looking at, say, the amplitude on one channel versus the amplitude on the other channel. And the different clusters correspond to the different neurons. Then you perform some sort of cluster analysis to group these into different, different groups corresponding to different cells, which then give you the times of your spike trains. And this has worked pretty well for about 10 or 20 years. This is what everyone's done, some variant of it. But there's an important thing to add, which is that you can't trust the algorithm to run perfectly. There still has to be a step of manual curation. And there are essentially 2 reasons for this. One is that the electrodes move relative to the brain, or the brain moves relative to the electrode. And this shows an example here of the amplitude of a spike on the y-axis versus time on the x-axis. And you can see that this spike is getting larger and smaller. And we're pretty sure this is because the electrode is moving. And I'll show you some data later that really makes that case very convincingly. And the algorithms can't always track this sort of drift, so it needs to be manually corrected. The other thing is that even if the electrode isn't moving, the cell can biologically change the waveform it generates. For example, the amplitude goes down in a burst or down after a period of intense firing for the neuron. And these things can't be modeled very accurately, at least so far, which means it's up to the user to use the information and their judgment to guess when something really is one neuron and when it's 2 neurons that have been merged together artificially or one that's been split artificially. So fully automatic clustering is not yet possible. At the end, I'll mention a possible way to get towards this. Okay, so does it work? When I was a postdoc in Yuri's lab, a colleague, Darrell Henze, spent several years collecting a data set of recordings from 4 neurons at a rate of about 1 neuron per year, which is still the most amazing data that exists to answer this question, where he had a tetrode wire tetrode outside of the cell and a sharp microelectrode that went inside the cell to record the action potentials with absolutely no error. And Yuri mentioned this data yesterday. Here we see the intracellular potential and the extracellular potential recorded on the tetrode. And the thing about this is it's ground truth. You really knew exactly when the cell fired because the intracellular waveform had no question. So we could use this to calibrate the system. And it shows the importance of manual curation. So what we see here is a cluster plot with, I'm sorry, this axis has been cut off. This shows the amplitude on channel 2 versus channel 3. And the different colors are different clusters of spikes. Every dot's a spike. The different colors are different clusters produced by the automatic algorithm. This is, at this time, a very old one called autoclass, but they all behave basically the same. And these have been artificially split because we know from the ground truth that this red elongated cluster here is a single cell which was recorded by the ground truth. And these 3 subclusters needed to be merged manually in order to get good performance of this. If you do that manual step, the ground truth says that the error rates for tetrodes are pretty good. The letters here are individuals who did what at the time was the common technique of manual clustering, and their errors were pretty bad, of the order 20%, 30% in some cases. Sometimes they were down to about 10%, but they were never very good. On average, the manual clustering was pretty bad. The red A's are the results of automatic clustering using this program autoclass, and those are usually of the order 5%. It's not perfect, but it's kind of okay. And the dashed and solid lines are the optimal performance you could have expected that we knew because we had the ground truth. And if you see the dashed line, it's a bit faint here, but it's normally right underneath the A. And what that's showing is that the automatic method, after manual curation, is performing about as well as it possibly could have. So that was all right for tetrodes, and that's why this is what people did for about 10 years or so, because it works basically about as well as it possibly could have, with a few exceptions. So here's an important exception. If we did the analysis restricted to these events called sharp wave ripples that I think you heard a bit about yesterday, very interesting, believed to be involved in memory processes and such, but you have a very large number of neurons firing synchronously, and if you compute the error rates only in that period, you see they're much, much worse. Even the best possible errors here are of the range 20% or so. And the manual spike sorting is now in the range of 50%. This is really a pretty bad error rate. It's not because the spike waveform has changed. This shows the spike waveform inside the ripple events and outside. They're nearly identical. But what's changed is the error rates, and in particularly the false positive errors are larger rather than the false negative. So it works most of the time, but the one time it really doesn't seem to work is when you have very synchronous firing, such as during these sharp wave ripples. Okay, so with tetrode, the traditional approach basically works. It needs manual curation. If you do that, you get to about 5%. It's worse at times of high synchrony, but the big problem is that this traditional method fails completely for larger electrodes, meaning eight channels is about the upper limit. By the time you're at 16, it no longer works. It fails pretty miserably. And as we heard yesterday, there are now new electrodes which have hundreds of channels. So this approach is a disaster for that. And we were part of the tester consortium for these probes. And Nick Steinmetz, who's over there, recorded the first data that we got from them a few weeks before the 2014 Society for Neuroscience meeting. This shows a segment of the raw data, time on the x-axis, channel number on the y-axis, and it's pseudo-colored, so that black means a negative potential and white means a positive potential. And you can see these little bumps which correspond to the times of the action potential, and you can see the shapes of them as well, which presumably are back-propagating spikes, and the negativity followed by the positivity. Beautiful data. So we got this data about a few weeks before the 2014 SFN meeting, and we were really keen to present it at the meeting. But doing a calculation showed that if we'd have used the old algorithm to process this recording, it would have taken 3.5 years to finish running. So that obviously wasn't going to fly, but that actually isn't the worst problem. The worst problem is that the manual curation would basically be endless, and also the estimated performance would be very poor. And the reason for that can be demonstrated here. This is a snippet of now older data, 32-channel data recorded in Yuri's lab. You see a spike here of one neuron, a spike here of another neuron. They're all spread over very large numbers of channels. Some of them are on smaller numbers of channels. This one's only on one channel. But the problem is that you have this phenomenon. You have a spike in one place at exactly the same time as a spike in another place. And the reason that that's a problem for traditional spike sorting is that it would extract everything at this time on all channels and try and do cluster analysis on it, not taking into account the fact that the spikes are local. And as the number of channels grows, the number of times that you have these simultaneous spikes on different channels tends to a probability of 1, which is why the estimated accuracy is 0. Okay, so how to proceed? And actually we did have a bit of a heads up on the problem. So we managed to modify the traditional method just a bit so that it resolves spikes overlapping in time, but not in space and time. So in other words, the situation you saw before, where you've got two neurons on different parts of the probe firing at the same time, that could be dealt with. But the case that you've got two nearby neurons firing at the same time, that cannot be dealt with by this modified traditional method. Also, the manual curation tool, which is still essential, had to be vastly sped up, leading to a program called Cluster Viewer that you can get here. It's now finally published about 2 years after development, just in time to be superseded by other techniques, which I'm going to talk about next. So the error rates with this method are pretty reasonable. They're back in the range of 5% or so, but it took days to run the automatic clustering, which is not a complete disaster. It's not like years, but it's obviously not what you really want. So in order to deal with the large data sets that we now have, there are two possible approaches. One is to modify the traditional approach based on spike detection and cluster analysis. And this is the system I just described is like that. Another approach, JR-Clust, that Tim mentioned yesterday, pretty much superior to ClusterQuick in every way. And there are others, and these all work basically the same way of extracting spikes and then performing a more advanced type of cluster analysis on it. But there's another approach called template matching, which works a radically different way, and this is what I'd like to talk about now. And two examples of programs that now work on this new data and work this way are Kilosort, which was developed by Marius Pachitariu in our lab, and Spiking Circus, fantastic name for a program. It's because when you write in Python, there's an actual official instruction that you, if possible, need to name your programs with some reference to Monty Python's Flying Circus, and that's how it came with this name. Anyway, a great name for a very good program. And the way that these programs work is they try and fit the original data with a mathematical model. So this shows in a simulation Nick made some data recorded as would be recorded from the probe, and the algorithm splits it into a mathematical model, which is a sum of template waveforms shown here for two cells in red and in blue at various times indicated by these spike times, plus a residual noise. So this residual noise is both electronic noise and the summed activity of all of the neurons in the background that are just too small to record. And you can see over here that the red and blue cell fire very closely together in time, and their waveforms have added, right? This is what could not be solved with the modified traditional approach, because even though it can deal with spatially separated, temporally continuous spikes, it can't deal with spikes that overlap in space and time, and that's what template matching at least we believe should be able to resolve, which is a way to get that residual 5% error down to something closer to 0. So I'm going to tell you a bit about Kilosort, which was developed by Marius Pachitariu. It's now available online. It's not yet supported, but if anyone wants to try it, talk to me or Marius, and we'll point you to the code. So what this does is it tries to optimize the fit of the model by reducing the size of this residual noise. It splits the data into a signal, which is the spikes, and a noise, which is everything else, and it tries to find the optimal spike waveforms and times to reduce the size of what's left. And it does that using a technique called dictionary learning, which is an alternation of 2 steps. First, given some template waveforms, you find the best spike times to reduce the error function as well as you can. And then second, given the spike times, you re-estimate the template waveforms, and you go back and forth between these 2 things until the algorithm converges, resulting in the maximum amount being explained by the signal and the minimum amount by the noise. So this shows it in action. This is how the algorithm is initiated with some things that look a bit like spikes, negative followed by positive, on all of the channels. This has nothing to do with the data, this is just the initialization. And what I'm going to show you is an animation of how the procedure happens. So what you'll see is that they evolve in time, starting to look a bit more like spikes. Most of the time the changes are small. Occasionally you have big jumps. You may be about to see one there. Yeah, and those big jumps are deliberate. They're to get it out of local minima that the algorithm gets stuck in. But after a few iterations, it finds some template waveforms that look like this. You've got negativities followed by positivities. You've got the sort of C shapes that you often see due to back-propagating action potentials going up the dendrite. And these are the template waveforms it's found. In principle, this ought to be very slow, because rather than just working on the spikes it's extracted, it's working on the entire data set, and it's going back and forth through the entire data set a very large number of times. In principle, this ought to be very slow, but Marius used a couple of tricks to make it actually way faster than the traditional ClusterQuick was. And the first is hardware acceleration. We're not talking about the sort of system you need to build to beat the world's champion Go player. We're talking about the sort of system you need to play Grand Theft Auto. It's a consumer-grade GPU card that you can get for a few $100s. There's actually a better one coming out pretty soon, and a solid-state disk. That's all that's needed. It's a consumer gaming system, and because it's programmed in CUDA, it runs much faster on the GPU. The second trick is a very clever piece of math, which is about how you compress this very high-dimensional signal into a smaller dimensional signal. So in black, you can't actually see very clearly in black because it's underneath the red curve. So let's just say in red is the original waveform of the spike. Sometimes the black one is visible underneath it. That's the real original waveform. And in blue and in red are 2 ways of approximating it. In blue, you're seeing the approximation of the spike waveform by a set of principal components which are the same for every spike. This is essentially the way the traditional method compressed the data by applying principal component analysis to each channel. And if you do that, you get the approximation in blue, which isn't always perfect. It's not that bad, but it's not always perfect. In red is another way of doing it, where you give each spike its own set of principal components. So it expresses the spike waveform on all channels as a sum of a small number of template waveforms. And the point is that although different neurons have very different waveforms to each other, for a given neuron, the waveform on the different channels is relatively similar. There's only a few degrees of freedom. So it's able to compress the hundreds of dimensions of channels to just about 3 dimensions per cluster. And what that means is that the algorithm time scales with the number of clusters rather than the product of the clusters times the number of channels. This is why it can run in a reasonable amount of time even on this process of looking at all the data. Another feature is an automated merging heuristic. So the manual curation process, where you have to merge the clusters after automatic clustering has happened, many of these merges are actually blindingly obvious, as anyone who's done this manual process will know. So Killasort has a few heuristics to take clusters like this that are obviously the same and to automatically merge them, saving the operator from having to do it. And this can probably be improved, and hopefully with time, the manual clustering time, which now is something like several hours to a day for a long recording, will be brought down to ideally nothing. Okay, and the final part is a new improved version of the manual phase. So in the same way that the early cars looked a bit like horse-drawn carts, the original manual software we had looked a lot like what you would use for tetrodes. But when you go to large count electrodes, there's some things that just don't make sense. With a tetrode, you really want to see all of your clusters at the same time. With a high count probe, that's completely impossible. So even trying to have that feature is a waste of time. So this program Phi, written by Cyril Rossin, doesn't even try and do that. It just presents the best two to the user, and then the user makes a decision on whether to merge them using things like the display of the waveforms and the autoencrust correlograms. And it's also much faster on these large data sets. So the system is able to make some pretty good distinctions. So here you see in blue and in red the spatial waveforms of two neurons recorded in one of Nick's recordings. The waveforms look almost identical. If you compare, the red one is just slightly shifted to the right. The waveforms look almost identical. There's a few places they're a bit different. And if you go over here, you can't see it that clearly. The mean waveforms are superimposed. You can see they're just slightly different. But we know they really are different cells because down here, you see the autocorrelograms of each cell showing a refractory period, as you would expect for neurons. But in the cross-correlograms, there's nothing. There's no common refractory period. This means it wasn't one cell that was artificially split in two. It was two cells with nearly an identical waveform. Had we not had this dense set of channels, we would not have known that this was this was two separate neurons, which is an example of how having these new advanced electrodes lets you do better science. We just wouldn't have been able to tell that before. Okay, so does it work? So the way to know if this sort of approach works is again using ground truth data. And I mentioned the data Darrell collected in 1998. Since then, there's been an addition, and he collected 4 cells. Since then, there's been an addition of 1 further cell collected in Adam Kampf's lab, which now is on I think a 32-site silicon probe. So that is progress, but it's nothing like what we need to test these algorithms. So we have to resort to simulated ground truth. And the way we do that is by taking a recording, taking a spike that was recorded in one part of the recording, and artificially transplanting it to another part of the probe. So we know exactly when the spikes are happening because we put them there. You can't quite literally cut and paste it, because if you do that, you'll copy not just the signal, but also the noise. So we need to clean it up by doing principal component analysis. What you see here in color is the original waveforms, and in black, you see a smoothed version, smoothed by principal component analysis. We subtract that from the original place and put it in a new place. And here, you see what's happened to the data set after we do this procedure. So here's the original data with the spike times in black for the negative and white for the positive, channel number on the y-axis and time on the x-axis. This is what's left after you subtract the spikes that had been manually clustered. You see some of them are left. This one, for example, is still here because that one that had not been manually detected. But this one is gone. We've subtracted it out. You can't see any remnant of it. If we hadn't have done that trick with the noise, you would still be able to see it because it would be exactly flat here. So the signal is gone, but the noise remains. And after that, we repaste it back in to a slightly different place, And what this has done is it's kept the amount of synchrony that you had originally in the data. So remember that the time that's worst for this spike sorting is when lots of neurons fire at the same time, close together in space. After we do this trick, this is preserved, because we've taken out the spikes and we've shifted them all down by 10 channels or so. So we know when they fired, where they fired, and the level of synchrony is exactly the same as it was in the original data. The relationships between the cells and the relationships between the cells and the local field potential is exactly the same. Okay, so what about the results? So this shows now for a database of 800 of these simulated ground truth neurons, we've got a cumulative histogram, sorry, not a cumulative histogram, a sorted histogram of the false positive rates here for ClusterQuick, the old method in blue, and KillerSort, the new method in red. And you can see the false positives are almost exactly the same. In fact, ClusterQuick does a little bit better for false positives because it rises more slowly. On the other hand, the false negative errors, the miss rates, KillerSort is doing way better because up to about neuron 500, there's essentially 0 misses, whereas ClusterQuick is doing worse because it's getting up to about 20% misses by that time. And the reason for this, we think, is the overlapping spikes. The spikes that overlap in space and in time that just can't be resolved with these modified traditional methods. And the total score is indeed better. If we now do a simulation of the manual stage, what the best manual operator would do, it doesn't change the results particularly much. ClusterQuick gets a bit better in the miss rates, but the total scores are still the same, that KillerSort is generally ahead. The other thing we can look at is the total number of merges required, which is an estimate of the manual operator burden, how long it would take to do this process. And you see that that KillerSort is ahead there. There are now about 5 algorithms competing, and this is a great situation to be in, where we have a choice of 5 to choose between. And this is an analysis Nick did comparing 5 of these. And you can see that in terms of miss rates, the 2 that are ahead are Spiking Sorcus and KillerSort, because these are template-based approaches. In terms of false positives, again, these 2 are maybe a bit better, not such a big difference. And in terms of manual merges required, JRClust is winning. And in terms of time, again, ClusterQuick is definitely the tortoise in the room, taking several days to finish running, and even weeks in the worst case, whereas KillerSort and JRClust are both under an hour, which is certainly an improvement on 3.5 years. So the results look a bit like this. After it's done, this is the raw data with the spikes detected, and that's, of course, just a small fraction of it. And as you zoom out, you start to get a sense of just how much data you're getting from this probe. This animation goes on for a bit of a while, but every colored dot is a different spike of a different detected cell. And time, of course, goes on much longer than just these few seconds we're showing here. And eventually, we will see the full length of this probe that Nick recorded from in an awake mouse. And the thing about this is that having this new technology, you can't help but do new types of science. So this probe, when we're interested in this area of secondary motor cortex, but we couldn't help but record from the striatum too. So now there's a recording of both brain areas. And here we see what you get with 2 probes simultaneously. This is, I believe, a new world record of the most neurons simultaneously recorded with extracellular electrophysiology, going through V1, hippocampus, and thalamus. And you can just already see that there's the interesting science here. You see the response to a flashed visual stimulus. If you look carefully in visual cortex, in the thalamus, you see something going on across thalamic regions. We don't know what that is. It's also there in the striatum. These may be movements the animal's making. They may be responses to sounds. We just don't know, but you can't help but do new types of science with this type of data. So just in the last couple of minutes, I want to talk a bit more about this point, about how new technology means you're going to do new science, and it isn't always what you thought you were going to do when the technology was first discussed. So the first one concerns fully automatic spike sorting, something that I'd always thought was pretty much impossible because of this problem of electrode drift. If the electrode moves away from your cell, it's gone. There's no way you can correct for that, except when you have a long linear electrode with regularly spaced sites. So this shows another of Nick's recordings with time on the x-axis, position along the probe in the y-axis, and the dot corresponds to spike amplitude. This was an acute recording which are particularly susceptible to drift. And you see there's these events where everything seems to shift. The black spikes have moved up, presumably because the probe has moved down relative to the brain, or the brain has moved up relative to the probe. And in fact, everything looks so consistent that it looks like it might be possible to spatially upsample and recenter the data to compensate for this drift. This is only possible because it's a long, dense electrode array, and it really hadn't occurred to us designing this probe that this would ever be possible. But if you do it, this is what you get. It looks still a bit jerky because this was just some manually drawn lines to estimate the position, but it looks like the drift hasn't happened. And in fact, if you now run spike sorting on this, it no longer has this problem of splitting that cluster into two. It detects it as a single event. So it may be that with this technique, and this data in particular, from a long linear electrode with regular sites, it will be possible to do fully automatic spike sorting. And that will be a tremendous saving of time and effort for all the scientists who spend all of this time doing manual curation. And the last thing I wanted to say is about another new type of science that you can't help but do with this sort of technology. And that comes from the fact that brain activity is global. So in neuroscience, we all tend to have focused on a single area. You know, I switched from hippocampus to auditory cortex. This is now a massive deal. You're switching from one region to another. But in fact, the brain works as a whole, and this is illustrated very well by some more data Nick recorded actually with a different technique of wide field optical imaging. So what you see here is a movie of a mouse brain in which GCAMP was in all cortical pyramidal cells. And you're seeing the mean fluorescence as a function of time. So red means that part of the brain is more active than baseline. Blue means it's less active than baseline. And this is a mouse that's sitting there looking at a screen, watching visual stimuli. And the interesting thing about this is I'd like to invite you to guess what part of this movie is the response to the visual stimulus. Was it that, for example, that big event? In fact, it wasn't. If we look here, you see that the stimulus comes on now, and if you look very, very carefully, you can see a slightly increased activity in this part of the left visual cortex, which is the response to this stimulus on the right-hand side of the screen. But nearly all of this activity that's going on had nothing to do with the stimulus we were presenting. It's coordinated activity across the entire brain, and you won't be able to understand it unless you can record from the entire brain, not just the cortex, but also the subcortical structures, the thalamus, etc., that coordinate all of activity. And that's what we hope will now become possible with these new technologies. Okay, so that's all I'd like to say. Thanks to the people who made this all possible, and of course the funders. Thank you very much.
Stimulating specific target neurons at the given specific wavelength. And when I take a look at this technology, and it looks like crude to me because most of the work has been done by sticking optical fiber into the